Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is for 15th December 2020. As all of us know, we are now not only completing the video tutorials, we have even completed the revision. I have given revision notes also to you. Now I am into the method of training you for your board exams. As part of the training, you know that your batch is getting a new style of question paper, and in that there is assertion and reasoning questions. So this is the reason why I am posting this video. Every chapter we are answering the possible assertion and reasoning questions with explanation, because unless you understand the concept, you cannot answer such type of questions. And as it is a reason, you need to reason out. There is no use of by hearting it. So do not by heart it. You have to understand it. So to understand it, you must be clear in your concept. Now with that background, let's move. Directions are same. You got 4 alternatives. Answer A, if both assertion and reason are true, and reason is the correct explanation of assertion. Answer B, if both assertion and reason are true, but reason is not the correct explanation of assertion. C, if assertion is true, but reason is false. D, if both assertion and reason are false. So, question 1, assertion. In a chemical engineering process, it is necessary to prepare sterile ambience. Reason. Sterile ambience inhibits the growth of undesirable microbes during manufacture of products like antibiotics, vaccines, and enzymes. Of course, the moment you read these statements, I am sure, being a 12th class biology student, you must have realized that this belongs to chapter number 11. Chapter number 11, Biotechnology Principles and Processes. So, when you read both assertion and reason, having understood the concept, I am sure you have no inhibitions. You will straight away put that A. Both assertion and reason are true, and reason is the correct explanation of assertion. Why? During sterilization, the equipments etc. are made free of any microorganisms before the addition of desired microbe or cell. Now this desired organism does not get any competition from other organisms and expresses all its characters and grows effectively. Hence, sterilization enables growth of only the desired microbe. It is also evident that along with genetic engineering, sterilization is also essential for successful biotechnological experiments. Question number 2. Asexual or asexual reproduction is more important with regard to biotechnology. Reason. Asexual reproduction preserves the genetic information, while sexual reproduction permits variations. So again, thinking of that, I am sure your mind goes for A. Biotechnology studies need a quick growing population of genetically uniform individuals, so that similar changes may be produced in all the genetically engineered organisms. Hence, asexual reproduction, which is a quicker and faster method of reproduction and preserves the genetic information of individuals, is better suited for biotechnological experiments. For example, if an organism that has been incorporated with a gene for disease resistance reproduces sexually, then recombination and segregation of genes will occur during gamete formation. Hence, some of the progeny may lose the gene of disease resistance incorporated into the parent. On the other hand, if the genetically engineered organism reproduces asexually, then all its progeny will invariably contain the gene for disease resistance. That's why biotechnology favors asexual reproduction. Question number 3. Genetic engineering can overcome the drawbacks of traditional hybridization. Correct? Reason: genetic engineering can create desired DNA sequences to meet specific requirement. This is also correct. So obviously the answer is A. The answer has to be A. Traditional hybridization procedures involve mating of organisms to be modified with another individual of the same species having desired characters and screening the progeny for expression of desired set of characters. These procedures very often lead to inclusion and multiplication of undesirable genes along with desired genes. Besides, interspecific hybridization are generally not very successful. Genetic engineering can overcome all these drawbacks because use of recombinant DNA technology, gene cloning, and gene transfer allow us to isolate and introduce only one or a set of desirable genes without introducing undesirable genes into the target organism, that too from any distant organism. So question number 4, assertion. A piece of DNA inserted into an alien organism generally does not replicate if not inserted into a chromosome. Reason: chromosomes have specific sequences called ORI. Remember ORI? Origin of replication. ORI reason, where DNA replication is initiated. Of course, again, the assertion is correct, reason is correct, and the reason is correct explanation of assertion. Origin of replication is a sequence from where replication starts by binding of polymerase enzyme. An isolated piece of DNA without origin of replication or ORI site cannot replicate and is gradually lost during nuclear divisions. But if it is inserted within a chromosome, it is replicated along with the chromosome as a result of replication process initiated by the ORI site of chromosome. This is the reason why recombinant vectors used in genetic engineering must contain origin of replication so that they can replicate themselves during cell division in order to ensure presence of recombinant gene in all the progeny. Now question number 5, assertion. Genetic engineering requires both nucleases and ligases. Reason: ligases produce the nick in the recombinant DNA molecule. I think the answer should be C. Yeah, why C? Nucleases are the enzymes that remove nucleotides or produce nick in the DNA strand. Exonucleases remove nucleotides from the free ends of DNA, while endonucleases produce internal nicks in DNA. Now the desired gene is inserted and then the cut ends are sealed with the help of DNA ligases. Ligases or ligases are also called molecular glue as they join together two DNA strands by forming phosphodiester bonds between adjacent nucleotides. Now question number 6. Restriction enzymes HIN, HPA are produced from two different genera of bacteria. Reason: HIN is produced from Haemophilus while HPA is produced from Hematococcus. Now what is the answer? Answer, let's see, D. Why D? In nomenclature of restriction enzymes, the first letter of the name of the genus in which given enzyme is discovered is written first in capital. It is followed by the first two letters of species name of the organism, and these three letters are generally written in italics like HIN and HPA. Both are produced from single genus Haemophilus, but from two different species. What are the species? It is Haemophilus influenzae and Haemophilus parainfluenzae. So this is how the answer has to be D. Question number 7. That is restriction enzymes recognize palindromic sequences. What are these palindromic sequences? Remember, I gave the word Gadag, G-A-D-A-G. I gave the word Malayalam. That means both forward and backward reading, the spelling is same. That is palindrome sequences. Reason, palindromic sequences read same in both directions of the two strands. So answer is to be B. And then question 8. Assertion. The matrix used in gel electrophoresis should have a controllable pore size. Reason. Agarose concentration can be changed to change pore sizes. Again, I think the answer should be B. Why B? The assertion and reason both are true, but reason has no correlation with assertion. That's why. And question number 9. All expression vectors are cloning vectors and vice versa. Reason. Expression vectors have at least the regulatory sequences, that is promoters, operators, ribosomal binding sites, and having optimum function in the chosen control but not origin of replication. I think both are false. Assertion is false, reasoning also false. So obviously the answer is D, right? All vectors used for propagation of DNA inserts in a suitable host are called cloning vectors. But when a vector is designed for the expression, that is production of the protein specified by the DNA insert, it is termed as expression vector. Expression vectors need regulatory machinery suitable for the host. That is, if an eukaryotic gene is to be expressed in prokaryotes, the expression vectors should contain gene without introns, because prokaryotes lack introns. Removal machinery for transcripts, but all the vectors including expression vectors must contain ORI so that they can replicate when the host cell divides and their presence is ensured in all progeny. Coming to question number 10. E. coli having PBR322. What is this PBR322? Plasmid, okay? With the DNA insert at BAMH1 site cannot grow in a medium containing tetracycline. Reason. A recognition site for BAMH1 is present in TTR, tetracycline resistant region of PBR322. I think it must be correct. So A. PBR322 has recognition sites for several commonly used restriction enzymes. Recognition site for BAMH1 is present in tetra-TTR region responsible for tetracycline resistance. When an insert is added at BAMH1 recognition site, the gene for tetracycline resistance becomes non-functional and the recombinant bacteria with the plasmid PBR322 that has a DNA insert at BAMH1 lose tetracycline resistance. Hence, answer A. Question number 11. Assertion. A bacterial cell with no restriction enzymes will be easily infected and lysed by bacteriophages. Reason. Restriction enzymes catalyze the synthesis of protective coat around bacterial cell that prevents bacteriophage attack. I think question number 11, answer C. Why C? Restriction enzymes were named due to the phenomena of host restriction of bacterial phages. Restriction enzymes produced in a bacterial cell recognize and cleave foreign DNA introduced, such as the bacteriophage, into the cell. Thus, bacterial cell cannot be infected and lysed by bacteriophage, and hence a bacterial cell lacking restriction enzymes is easily susceptible to infection of phages. The DNA of the host bacterial cell is protected from its own restriction endonucleases by methylation, usually of A and C, within their recognition sites. Hence the answer C. Question number 12. Assertion. Special methods are used for transformation, that is, incorporation of rDNA into host. And the reason, DNA is a hydrophilic molecule, of course, without any doubt. 12a. Transformation is permanent heritable change in a cell, particularly in a bacterial cell, that occurs as a result of its acquiring foreign DNA. But cells normally do not take up foreign DNA molecule because DNA is a hydrophilic macromolecule and its movement through lipid membrane is opposed. Special methods such as CaCl2 treatment, electroporation, particle delivery method are used to make host cells competent, that is, able to take up foreign DNA. Question 13. Use of chitinase enzyme is necessary for isolation of DNA from yeast cells, but not in case of Spirogyra. Reason: fungal cell wall is made up of fungal cellulose or chitin. Of course, without hesitation, it has to be A. Release of DNA from a cell is achieved by treating the cells or tissues with enzymes such as lysozyme, that is bacterial enzyme, cellulose, plant cells, chitinase fungus. These enzymes degrade cell wall. Plasma membrane degrading enzymes like lipase are also needed. Since yeast is a fungus and fungal cell wall is made of chitin, isolation of DNA necessarily requires use of enzyme chitinase. That's why it has to be A. 14. Assertion: PCR, polymerase chain reaction. PCR primers must not have self-complementary reasons. Reason: self-complementary reasons result in hairpin structures adversely affecting the PCR. So it is true. Primers are nucleotide sequences that serve as base for formation of new DNA strand. In PCR, primers are added on the ends of DNA strand to be copied, and the primers are extended towards each other so that the DNA segment lying between the two primers is copied. If self-complementary bases are present in primers, then hydrogen bonds are formed between them and hairpin-like structure is formed. It makes the primers unsuitable for extension of strands in PCR. Now come to the last question, question number 15. Downstream processing is generally considered more difficult and costlier in plants than that in microbes. Reason: rhizosecretion is used as a method to facilitate easier recovery of recombinant proteins from plants. So the answer should be for 15 B. Why B? Assertion and reason are true, but reason is not correct explanation of assertion. As you know, we are talking in assertion downstream processing, and in reason we are talking something else. That's why there is no reason, but both the statements are correct. So this is how you need to understand the concept. Based on understanding the concept, you can answer all the questions. So keep on practicing. This will fetch you very good marks in the board exam.